Javier Parladé is of the Research and Technology Food and Agriculture Institute of Catalonia. Is a guest speaker of this session on biology and population genetics. He has participated at a lot of numerous national and international projects on application of ectomycorrhizas. Now is involved in cultivation of edible ectomycorrhizal mushrooms, and as you can see here in his presentation, is involved in quantification of mycelium. Tuber mycelium means soil. Please.
Thank you, Alessandra. Well, good afternoon to everybody. First, I wish to thank the organizers for inviting me to participate in this Congress. I'm going to talk about the quantification of mycorrhizal fungal mycelium in the soil and the methodology associated, focusing the real-time PCR, the specific traceability of ectomycorrhizal edible fungi, and the results we have with TUBER in the quantification of soil mycelium and in the detection of the mating type genes by comparative CT. And we'll finish with concluding remarks. The ectomycorrhizal symbiosis has been studied mainly by direct sampling of sporocarps and mycorrhizas, and then with morphological and molecular identification. In turn, the exarradical mycelium is the hidden part of the symbiosis and has been studied mainly by indirect sampling and analyzed by molecular techniques. The mycelial systems of ectomycorrhizal fungi play a critical role in plant soil translocation of nutrients and water, in the mobilization of nutrients, organic nutrients, phosphorus and nitrogen, and has a possible effect on carbon sequestration. Soil is the second largest carbon sink in the planet after the oceans. And interestingly, the mycelial systems are indicators of the spatial temporal fungal persistence in the soil. The detection of extraradical mycelium in the soil can be done by different methods. We have fungal biomarkers like ergosterol and fatty acids or phospholipids, which are components of the membrane of the fungal cells, and the chitin, which is a component of the cell wall of the fungi. They are estimators of the fungal biomass and that are non-specific. We also have genetic markers based on the DNA analysis, and with these markers we can study different levels, at community levels, species levels, or even strain level. All these genetic methods are qualitative and mostly based on the PCR. The conventional PCR is an enzymatic reaction consisting on the exponential amplification of a region of DNA region of interest. When we run this reaction, in each cycle, we double the amount of products until we reach an amount to be stained and to be visualized in a gel. This is the shape of the amplification curve in a PCR with an exponential phase and then a plateau phase indicating that the regions run out. If we have different samples, the analysis in this area, in this plateau area, we cannot differentiate between these different samples, but they can start at different points, so we cannot quantify with this technique. To solve this, we have a technique called real-time PCR, which can be applied with two types of chemistries. The cybergreen chemistry uses a dye called cybergreen, which intercalates and binds in double-strand DNA and then emits fluorescence. With a fluorescent reader in a thermocycler, we may have the amplification curves in which we can follow the amount of DNA which is proportional to the amount of fluorescence detected. This system has the problem of inspecific amplifications. This may be detected by the melting curve. If we have some contamination, we have also a signal of fluorescence. So if we, at the end of the process, increase the temperature, we can calculate the melting point, which is represented here, and if we have more than one peaks in the melting curve, this indicates that we have more than one product amplified. And this may be a problem because we can overestimate the final quantification. So the advantage of this methodology is the price. The reasons are relatively cheap, and the drawbacks are the specificity. So we have another alternative, which is to use the Tagman probes. The Tagman probes are specific probes which anneals to one strand between the two primers. Then we have three specific points. When the polymerase extends, the quencher in one end of the probe inhibits the fluorescence in the other end from the reporter. But when the Tag polymerase arrives to the probe, degrades with its exonuclease activity, degrades the 5' end of the probe, hydrolyzes it, and then the reporter is free. And when it's free, this reporter emits the fluorescence. And then the increase of fluorescence, similar with the cybergreen, is proportional to the amount of product formed. Well, this is the opposite to cybergreen. The advantage is the high specificity, and the drawback is the price. These probes with the fluorophore are quite expensive. Well, these are the standard curve fundamentals. We start from known quantities of DNA, and here we can see the different amplification curves. We, in these curves, with different dilutions of known amounts of DNA, we can establish a threshold line here, and then calculate what we call the threshold cycle, or CT. This is an important parameter because with different points in which we cross this threshold, we can make the standard curve. The standard curve plots the CT values and the starting quantity of DNA. This standard curve, if has an efficiency of 100%, means that in each cycle, the amount of DNA doubles. It is allowed for a good reaction that the efficiency value, which is calculated from the slope of this curve, is between 90 and 110%. If we have a sample, an unknown sample like this, what we do is to calculate the CT of this sample and interpolate this in the standard curve. So we have the initial amount of the DNA of this soil sample. So the protocol for the soil mycelium quantification we use in our lab starts with selecting a specific target sequence, which is always in the ribosomic DNA region, the ITS region, which is specific. And then in this region, we design the primers and the probe in the case of the Tagman chemistry with the appropriate software. Then we prepare the standards, weighting the mycelium from target fungal cultures in the fungus which we can culture. If not, we can use sporocarps. Then we mix a known amount of mycelium with soil free of the target fungi. This is to compensate the possible effect of the soil inhibiting the PCR. Then we extract total DNA from the samples, dilute the samples n-folds to establish the standard curve. Then we extract also the DNA from the soil samples, perform the real-time PCR of the samples and the standards, and then interpolate the samples in the standard curve, as we have seen before. So far, we have designed several probes. These are the ones which are published with different species of mycorrhizal fungi, edible mycorrhizal fungi. Lactarius deliciosus, Rhizopogon, 2 species, Boletus edulis and Tuber melanosporum. We are working with Amanita caesarea and Amanita ponderosa prof now, and we are about to start an experiment with Terfisia claveri. The methodology for soil sampling is relatively simple. We use a metallic probe, and we take the first 20 centimeters of soil. The problem is where and when to sample, because of the soil heterogeneity and the patchy distribution of the mycelium in the soil. It's obvious that it's not the same, take a sample here or here. The mycelial distribution in the soil depends on several factors, physical, chemical, and biological, as the soil density, the biological interactions, the available nutrient resources, and the pattern of exploration types of the ectomycorrhizas. In spite of these problems, which generate a lot of variability, we have some results on the specific persistence of several fungi, which I'm going to explain. In field plantations, we have found in plants inoculated with the saffron milkcap, Lactarius deliciosus, positive correlations between mycorrhizas and mycelium, and we have studied the competition relationships with Rhizopogon species. In the case of productive forests, we have worked with Boletus edulis, with the king bolete, and with the Lactarius deliciosus too, in collaboration with the teams in Soria. And we have found that this real-time PCR is 100 times more sensitive than the conventional PCR to detect the mycelium in the soil. We observed in both species a peak after the fruiting season, a peak in the biomass of the mycelium, which more or less is in the winter season. And we haven't found a relationship between the soil mycelium and the historical productivity in sporocarps of the plots. In the case of Tuber melanosporum, we have a recent experiment which was carried out in collaboration with Ana de Miguel and Raimundo Saez and Herminia de la Varga from our lab too, to quantify the extraradical mycelium of Tuber melanosporum in several truffle orchards in Navarra. The objective of this work was to compare the black truffle mycelium biomass in different plots which had different historical productivity and watering regime. The 5 plots sampled were productive, watered, productive, non-watered, non-productive, productive of tuber brumale, and a natural area next to the orchards. So the soil sampling consisted of taking 4 subplots per plot of 100 square meters, and we took 5 soil cylinders per each subplot, which makes 20 samples per plot, and 100 samples for a total of the 5 treatments. This is the picture of a natural area in which we made the soil sampling in the Sierra de Lóquiz. And the results are presented here. This is a standard curve with the detection limits. The detection limit for the last dilution was 1.48 micrograms of mycelium per gram of soil. We found more mycelium, 10 times more mycelium in natural areas. This was surprising for us because in the orchards it's supposed to be cultured to favor the development of the mycelium, but we found much more mycelium in the natural areas. We found less mycelium in the irrigated orchards. Maybe there is a competition relationship that we cannot control. And we also observed a coexistence with the plots producing tuber brumale. We found also a mycelium of Tuber melanosporum. And we have to take into account in this experiment the possible influence of the sampling date. In this case, we have not the result of the seasonal variation of the mycelium as we have in the case of Lactarius and Bolete. So to review the studies done in the last 10 years on the use of the real-time PCR on mycorrhizal soil mycelium research, we found about 30 papers which used more or less equally both techniques, Tagman and Cybergreen, and these are the references. And in the case of the TUBER studies, we have several studies on TUBER-Borky, on gene expression, which is to quantify the complementary DNA to the RNA expression. And then with TUBER magnatum, one study of soil mycelium detection by Tagman probes, and both types of studies for gene expression and mycelium detection in TUBER melanosporum. So the recent advances in molecular biology, especially unveiling the genome sequencing and the biological cycle of TUBER melanosporum, led us to study the, no, sorry, led us to sorry, not, to bring us to the heterotalism of TUBER melanosporum. So now TUBER melanosporum is heterotalic. Sorry, this is a little mess with the English, and the location of the MAT genes responsible for the two mating types discovered. So using this information, we applied the real-time PCR to quantify these MAT genes by comparative CT. The objective of the study was the relative quantification of the MAT1 and MAT2 genes in the mycelium of TUBER melanosporum from soy DNA by comparing the CT values. The protocol was first to design the MAT primers and probes with Tagman probes in this case. We had a first test with cybergreen, but was not specific. Then we established the endogenous control or normalizer, which is a region which is expressed or present in all the samples. So if we want to distinguish MAT1 and MAT2, we consider that the ITS region is present in all the samples. So we used the primes and the probe described before for mycelium quantification. And then we established two reference samples, single mycorrhizas of TUBER melanosporum belonging to either MAT1 or MAT2 mating type, which was previously determined. So we have experimented with different samples. This is a preliminary study. The negative samples were the PCR blank with water, soil from Pinus sylvestris, in which it's not supposed to have TUBER melanosporum, soil from Pinus pinaster forest, both in Soria, 3 ascomycetes, Prosporella brunea, TUBER brumale, and Terfisia olbiensis, and 1 basidiomycete, Boletus edulis. And the theoretically positive samples were the 2 mycorrhizas used as reference samples, the mycorrhiza MAT1 and the mycorrhiza MAT2, 1 sporocarp and TUBER melanosporum, and different samples of soil taken in the orchards in the previous experiment. And here are the results. In the comparative CT results, we have a nice amplification in the detection range, as expected, of the ITS1 region. And in the case of the reference samples, we also had, in the case of the amplification of the MAT1 gene, nice amplification of the mycorrhiza M2, and in this case of the sporocarp. And in the case of the MAT2, we had the amplification very displaced in the limit of the detection range to the right, but we had nice amplification of the mycorrhiza belonging to the MAT2 compatibility type. These are the threshold lines indicating that the detection range are okay for this region. This is a multi-copy gene, and the production is higher than this single copy gene, but we have nice amplification here for mycorrhiza and the sporocarp, and here the soils, which are here, had very displaced amplification. All the negative samples gave no amplification. To make comparable the CT experiments, we have to compare the PCR efficiencies of all the reactions, and they have to be equivalent or differ in no more than 5%. So to do that, what we did was to test the ITS probe or endogenous control. Yeah, it can be hard to see, but I can explain you. And making dilutions of DNA from the sporocarp. Just 5-fold dilutions to make the standard curve. These are the dilutions, supposed to be the dilutions for the MAT1 sample, which won the mycorrhiza, which was known to be MAT1, and then the MAT2 probe tested with the mycorrhiza, which was MAT2. And then when we compared the slopes here, we found that all the slopes were very similar, so the efficiencies were very similar too, and the cities were comparable. So these are the results of the relative quantity of the different MAT genes in the soil samples and here in the mycorrhiza samples. These are the reference samples, which are 1, and this is the sporocarp sample. And all of the other samples are soil samples. The results indicate that from the 16 soil samples analyzed, 7 were MAT1, 4 were MAT2, 2 had both compatibility types, and 3 sample soils had no detection of these mating genes. There was no apparent relationship with productivity. In these preliminary samples, we had that no amplification in this non-productive plot. Here we only found the MAT2 genes, and here we found both of them. Also, both genes are in the limit of the detection range because, as I told you, they are single copy and difficult to detect compared to the ITS. MAT1 genes have lower CT values. 60% are under cycle 35, considered a little bit the limit of the detection range. And in the MAT2, all the amplifications were in a cycle beyond the cycle 35. So, but since the comparison of the efficiencies are similar, we expect that an increase of the initial amount of template, which are the experiments we are doing now, the number of replicate for the MAT2 experiments, and the optimization of the PCR conditions will refine these results in the near future. So to conclude, we know a lot of things in the biology of truffles that were unimaginable some years ago, but we still don't know, or only partially know, several things: how to sample the soil, the seasonal mycelium distribution, the mycelium responses to cultural treatments, the relationship with mycelium, soil mycelium and fruiting, the distribution of mating type genotypes, and the soil interactions which occur in the soil. The presented research has been done in collaboration with all these researchers belonging to public and private universities and research centers from Spain, and the research has been financed by the Spanish Ministry of Science and Innovation. So I want to thank to all of them for their collaboration, and thank to all of you for your attention.
Thank you very much, Javier. The next presentation is of Antonietta Mello from CNR of Torino, Italy.
So, good morning. I will speak about integrating approach to evaluate the productivity of tuber melanosporum in a model plantation. I want to say that it is really a challenge to study the ecology of a fungus in the soil because we know the distribution of an ectomycorrhizal fungus is very difficult to be followed. Okay. And for this reason, studying this soil ecology needs to take into account methods and strategies that can consider all the complexity of an environment such as the soil. So, to set up a protocol to check the fertility, or to predict the fertility of a tuber melanosporum ground, I mean, is really difficult, and we should start to consider. Is really difficult, but could be really helpful for truffle operators that want manage their orchards and can predict the yield of tuber melanosporum before the arrival of the tuber melanosporum fruiting bodies. So, before starting to speak about this protocol, I just want to briefly summarize what we know about the tuber melanosporum production. So we know that the truffle fruiting bodies are usually collected in the burned area. Everybody knows in this audience that is an area devoid of vegetation near or around the host trees. So moreover, what we know that this area is an expansion year by year, and this expansion causes deep changes in the roots of plants, in the hormonal balance, and proceed toward a very aggressive area that is the brûlé periphery. So in this area, there are expected to be very important changes also in light exposure and in moisture levels. So we have seen this morning a presentation about, peculiarities of this area from the chemical, physical features of the soil. This niche, because it is a peculiar niche, is affected by tuber melanosporum metabolites. So tuber melanosporum diffuse toxic metabolites that are recently recognized as allelochemicals. Many allelochemicals are present in soil, and phenolic compounds represent an important group of these chemicals, and they are high concentrated in the brûlé. But the higher concentration is just at the demarked area that is the most aggressive area of the brûlé. So this is a demonstration, in my opinion, of the close relationship existing between tuber melanosporum allelopathy and brûlé formation. So, which other factors affect tuber melanosporum production, tuber melanosporum fruiting body, and how they are correlated to the brûlé formation? Surely, among other factors, an important driving factors are changing of fungal populations. Okay, recently, thanks to the environmental genomics applied in soils collected in brûlé at Cahors in France with the help of Pierre Sousard, we have demonstrated that tuber melanosporum has an inhibitory effect on the other ectomycorrhizal fungi. So it decreases the quantity of other ectomycorrhizal fungi in the brûlé, so has a competitive effect. And in this context, also recently, we have seen from the sequencing of the tuber melanosporum genome in which we have been involved, we have seen that two metin-type genes are involved in the reproductive process. And now we are able to follow their distribution. We have seen from the previous presentation their distribution in a plantation. So, I think that I can say that we have to take in consideration all the context in which the research at the moment moves. In order to set up a method to check the fertility of tuber melanosporum, we thought to consider at least 3 factors. To see if there is a correlation between the abundance of tuber melanosporum in soil, the presence of mating type genes, and productivity. In order to reach this goal, we considered a plantation in Italy, in Piedmont, that is considered a model plantation since it presents 3 particular areas: a productive area, a non-productive, and an ex-productive area, an ex-productive area where there are ex-productive ox trees. So we started from the hypothesis that different sites could show different amount of tuber melanosporum, and we wanted to see if there is a correlation between this amount and the productivity of trees, the productivity of truffles. Okay, sampling was made in 2 periods of tuber melanosporum life cycle, in April and in October, and under 2 different host trees, Quercus pubescens and Corylus averrana, to see if these 2 conditions could affect the productivity and the distribution of mating type genes. So, from the experimental point of view, the quantification of tuber melanosporum was carried out by using primers developed by Bonito et al. In an assay of quantitative PCR specifically set up in our study. Whereas the survey of the 2 mating types genes was carried out by using primers already developed by Rubini et al. So, if we look at this scheme, we see that the 2 mating types were both found in the plantation, indicating that this is fertile, and that one mating type, the plus, that indicated with the plus sign, was more frequent in the area, whereas the other one was found only in a sub-area. And this data is in agreement with other data already published by Rubini et al., because the 2 mating types are not equally distributed in a plantation. We still have to understand why. And only a sub-area showing one of the 2 mating type could have the second one at a distance, at a minimal distance of 60 meters. Looking at a correlation between all the factors we considered, you don't need to look in detail at the table, but we just here reported some of our plants that considered in the study, so the productive, the ex-productive, and the non-productive, the 2 periods of sampling, April, October, results from the quantification and results of the survey of tuber of mating type, and we see that we could not detect the mating type under unproductive trees, the ones marked in orange, where the tuber melanosporum quantity was very low, under a threshold limit, 0.3 nanogram. Okay, and this is easily explained by the fact that we considered different locus. The one is ITS, is a repeated locus, and the other one is a single locus. So it is explained from the experimental point of view. And now we see which results we have had in relation to host plant and season. In April, tuber melanosporum DNA concentration was different between the productive and the non-productive sites. I mean, was higher in the productive sites, but not between the oaks and hazelnuts. Whereas in October we could not get sufficient data for the non-productive sites, so we could not make this comparison. And the tuber melanosporum DNA concentration was different between the oaks and the hazelnuts. So this last result could be explained by the fact that in October the life cycle, the truffle begins to be produced. So it's a question of the life cycle. And the difference we found between the two host plants probably is connected with the fact that Corylus avellana has a different apparatus, more wider and broader than that of oaks, and so can take contact in many places with the mycelium of tuber melanosporum. And from this mycelium, the growth of the truffle starts, and also in October, the truffle, I can say, as is a spore, of course, and also these spores concur to the production of DNA, the one we quantify. Of course, this data should be confirmed on a larger number of samples. So at the moment we have just a protocol, but it needs to be applied on a larger scale.
Yes,
what I can conclude from this research, the work highlights the limits of a well-defined protocol in order to check the fertility of a tuber melanosporum plantation. So, first point, the detection and quantification depend on the primer pair used. We have made many tests and the results can be slightly different. We have seen that mating type detection is possible above a threshold value, and in any case we suggest in order to obtain knowledge on the fertility of a ground, to combine all these approaches. I want to just remind you that up now the establishment of a plantation was based exclusively on data coming from the chemical physical parameters of soil. Now, we have more possibilities with the molecular techniques, and I think that truffle operators who want to use these techniques can look at trained companies such as spin-off of university or other services that can make PCR, in any case, can help the truffle operators to manage its truffle ground or its orchard before the arrival of the fruiting bodies, because we know that the period from the starting of the procedure to the collection of fruiting bodies is really long. So it is expected that the owner of a truffle ground, of an orchard, wants to know if there is the mycelium in the soil, is this orchard is potentially productive. By the way, we know that many factors contribute to the production of truffle fruiting bodies, so abiotic and biotic factors, and probably the complex of so many factors is the case for the heterogeneity of tuber melanosporum yields that remains highly unpredictable.
Okay.
So,
Thank you very much,
thank
Tonia.
you.
Thank you very much, Antonietta. Now we have a small change in the program. the two presentation of Carlos Colinas and Christine Fischer are inverted. So now we have Carlos Colinas presented. Carlos? Okay.
Gracias, Sandra.
Okay, so.
Y gracias también al comité organizador por la oportunidad de compartir este trabajo con todos vosotros. A ver si me aclaro con la presentación. Gracias. En este trabajo hemos intentado usar... Intentar ver un poco cómo se mueve el micelio de tuber melanosporum en el suelo, cómo se disemina, hasta dónde llega. Y lo primero que quería comentaros es que ese trabajo es un capítulo de la tesis de Lubin, que desgraciadamente no ha podido venir para el Congreso, aunque era su intención, y os manda recuerdos a todos. ¿No sería fantástico? Cuando Salvador Dalí pintaba este cuadro era justo la primera época en la que la gente empezaba a trabajar un poco en serio todo el tema de truficultura. ¿No sería maravilloso poder ver las trufas levantando la piel del suelo? Es un poco el sueño detrás de este tipo de trabajos, ¿no? Podemos intuir por dónde se mueve el micelio, podemos ver quemados que tienen un tamaño u otro, una edad que casi nunca sabemos la que es en la naturaleza. Podemos tener idea de un quemado cultivado y ver que a lo mejor pues una plantita muy pequeña nos hace un quemado muy grande y pensar que todo esto es micelio o no. ¿Y cuánto? ¿Es homogéneo o hay una variabilidad dentro del quemado? ¿Realmente se para el micelio en el quemado o continúa más lejos? Todas estas son las preguntas que realmente nos preocupaban. Entonces la idea fue intentar establecer una correlación entre la cantidad de micelio de tuber melanosporum y la edad de la plantación, porque claro, en un bosque, la edad de un bosque es indeterminada, en estos bosques mediterráneos de Recepe. En una plantación sí que podemos saber que hasta que se plantó ese árbol con trufa no había allí ni árbol, ni trufa, ni nada, era un campo de cereal. Y luego también la distancia. Y como íbamos a tener las muestras, también nos planteamos como objetivo el intentar ver si había brumáleo o si había el riesgo terrible de que hubiera indicum en alguna plantación. Establecimos una cronosecuencia con plantaciones muy jovencitas, menos, hasta plantaciones de 20 años, en total 6 edades, 3, 5, 7, 10, 14 y 20, un poco más espaciadas hacia el final, con 3 plantaciones por edad, para que esto pudiera tener un poco de consistencia estadística. Entonces, muestreamos el tipo de árbol dominante en la plantación. O sea, en las plantaciones de 3 años ni hay quemados ni hay producción. Entonces, muestreamos árboles que no tienen ni quemado ni producción. En edades intermedias, si la plantación era una plantación que ya estaba en producción, usamos árboles en producción. Si era una plantación que dominantemente ya tenía quemados, aunque no tenía producción, usamos los árboles con quemados sin producción, no el único árbol que quizá ya tenía producción de la plantación. Y tomamos muestras a 3 distancias, a 40 centímetros del árbol, a 100 centímetros del árbol o a 200 centímetros del árbol. Quitándolos, y 3 muestras en cada círculo que después compusimos en una única muestra para evitar la posible variabilidad que pudiera haber entre una parte botada quemado, norte, sur, etc. Quitamos los 5 centímetros superiores para quitar pues, hojarasca, detritus, y cogimos 25 centímetros de profundidad y un cilindro de 7 centímetros de diámetro, que cribamos en el campo directamente por una criba de 7 milímetros para eliminar pues eso, ramas, raíces, etc. Entonces, de estos suelos extrajimos DNA, de las muestras de suelo con el Power Soil DNA Isolation Kit de Mobile, y para controles y para muestras positivas de esporocarpos de Melanosporum, Brumalia indicum con el EasyDNA Fungal DNA Mini Prep Kit. La amplificación usamos el multiplex de Francesco, y para las muestras para la cuantificación de melanosporum usamos solo los primers específicos de melanosporum. Y esto no se ve muy bien. Abordamos la cuantificación, en vez de con la RT-PCR, con un estándar. A ver cómo lo explico en medio minuto, que es todo lo que tengo. Por una parte, establecimos una serie de diluciones de esporocarpo de tuber melanosporum, con una cantidad de tuber melanosporum conocida en una solución. Por otra parte, comprobamos que en los suelos con los que estábamos trabajando no había inhibidores de PCR. Y comprobamos esto utilizando distintas concentraciones de suelo con una concentración conocida de DNA de tuber melanosporum. Si hubiera habido inhibidores, los productos de amplificación se habrían reducido al aumentar la cantidad de suelo. Y después, en el mismo gel, poníamos nuestro estándar y después un bloque. Las 3 muestras de 3 distancias, de 3 años, de 5, de 7, 10, etc. Entonces, esto hicimos el análisis de densidad de las bandas obtenidas, siempre trabajando en la parte exponencial de la curva que nos presentaba Javier hace un rato de la amplificación de PCR para evitar la saturación. Entonces, las bandas, o sea, la densidad de banda del estándar serían los puntos rojos, que nos permitía hacer el ajuste de una curva, y a partir de la densidad de esta curva y de las densidades de las bandas observadas, podíamos estimar la cantidad de micelio de tuber melanosporum que había en esos suelos. Uno de los resultados más importantes que observamos fue que no observamos ni una sola muestra con tuber brumale. Bueno, quiero aprovechar esta ocasión para felicitar a los truferos de Teruel, que es donde están todas estas parcelas, por su capacidad de gestionar las plantaciones lo bastante bien como para que no haya brumale en ellas. Y afortunadamente tampoco encontramos indicum en ningún sitio, lo que fue un respiro. Vimos que tanto la distancia como la edad tenían un efecto significativo sobre la cantidad de micelio de tuber melanosporum en el suelo, así como la interacción. No tan fuerte, pero había una significación. Entonces lo que hicimos fue separar todos los tratamientos como tratamientos independientes, y al analizarlos de esa manera las diferencias eran francamente importantes entre las distancias, edades, etc. Resultados. Muchas barras, pero vamos a ir por partes. Tendríamos por una parte las 3 distancias en cada una de las edades, y por otra parte los miligramos de micelio estimados a partir de este estándar que construimos antes. Vamos por partes. Vamos a considerar primero la edad más joven, las plantaciones de 3 años. A 40 centímetros encontramos una cantidad más alta de micelio. Y ojo, porque esta escala es logarítmica. A 1 metro encontramos 1000 veces menos micelio. Y a 40 centímetros, casi 100 veces menos que a 1 metro. O sea, una caída importante en esta primera edad. Vamos a pasarnos a la segunda edad, 5 años. Estamos hablando de plantitas así, ¿vale? Ocasionalmente alguna puede producir alguna trufa, pero son plantitas muy pequeñas. Más o menos la misma cantidad a 40 centímetros, pero como 100 veces más a 1 metro. En muy poco tiempo parece que el micelio realmente se propaga. Y a 2 metros, otra vez, la planta levanta medio metro, a 2 metros de distancia ya tenemos una cantidad más que apreciable de micelio. Si ya miramos las demás edades, no hay diferencias. A partir del 7º año, curiosamente, cuando empezamos a ver las primeras fructificaciones, la cantidad de micelio ya permanece constante para los años y para las distancias. Vamos a mirarlo ahora desde otro punto de vista, y que es la que se alcanzaba desde la primera edad. O sea, a 40 centímetros, que no hay diferencias entre ninguna de las edades, siempre la misma cantidad. Mirando otra vez distancias, vemos que en los 40 centímetros sube bastante rápidamente y luego se estabiliza. Perdón, en los 100 centímetros. En los 200 centímetros sube no tan deprisa, pero también se estabiliza. Así que estos son más o menos los resultados y quisiera dejaros con una pregunta. ¿Tenemos una capacidad de carga para TUBER melanosporum en el suelo, de la que nunca se pasa? ¿Es constante esta capacidad de carga para todos los sitios o depende del sitio? Depende del suelo, depende del agua, depende de la especie arbórea con la que está. Yo quisiera rogaros a todos que hagáis muchos estudios de estos, porque ahora lo excitante sería saber si esta cantidad es más o menos la misma en todas partes o de qué depende. Y si realmente hay que esperar a llegar aquí para empezar a tener producción o no tiene nada que ver con la producción. Y nada más. Muchas gracias. Además de con la duda, querría terminar con un agradecimiento especial a la Asociación de Truficultores de Teruel por toda su ayuda para encontrar todas estas parcelas y buscar todas estas muestras, y también a la agencia que nos financió el estudio. Muchas gracias.
Thank you. Thank you very much, Carlos. The next speaker is Paul Thomas of Mycorrhizal System of UK.
Thank you. Okay, thank you. I'd like to talk about the survival of Tuber melanosporum mycorrhiza in suboptimal climatic conditions, because we've heard a lot over this conference about melanosporum in optimal climatic conditions, so this will be a little bit different. And when I refer to suboptimal climatic conditions, I'm specifically referring to a research project we have in the UK. Okay, to start off with, I'm going to talk about the geographic extent of Tuber melanosporum, only very briefly, and then about the climatic considerations, what we already know, and then talk about how we've gathered the data. And we've done two things. We've gathered data from a wide range of producing sites and amalgamated that data. Then we've taken data from suboptimal conditions in the UK and overlaid that data. So I'm going to talk about those two data sets, and then obviously go through the results, including at the end of the results will be some correlations within those suboptimal conditions. So first off, this is a map of the geographic spread of Tuber melanosporum. The red highlighted areas are where it's pretty much dominant in Europe, where the bulk of melanosporum comes from. Of course, we've got records from other countries on here, such as Germany, but this is where the bulk of wild melanosporum is. The green highlighted countries is the anthropogenic spread, really, so it's where we've taken it out of Europe. And these aren't all producing sites, but these are sites where there's trees in the ground and the mycorrhiza are surviving. So as we've heard, it's in China, Australia, South America, and very excitingly, actually, heard today in Gotland from Christina Whedon, and Austria with Alexander Urban. So that's fruiting sites at really high latitudes, which for me is very exciting. So what we know about the climatic conditions already is basically we need a climate with well-defined seasons, not characterized by extremes. Warm summers, high temperatures can be tolerated with the right kind of management. Prolonged cold snaps in the winter can be detrimental, but still we need relatively cool winters. And rainfall patterns which are relatively well spread. So we need rainfall in the early part of the year to trigger sporocarp initiation, and then rainfall later on in the year to help with maturation. And then this kind of basic climatic data has been used in a whole wide range of studies to look at new areas to grow truffles, and that's still ongoing. For example, this is a report from back in 1996, focused on Australia again. But there's few studies where they've monitored Tuber melanosporum development of the mycorrhiza outside of its climatic optimal. So that's what we've done here. So the methods. The first one to gather the wild type data. We took data from published materials, so from peer-reviewed research articles where they had highlighted fruitification. So all these sites were fruiting, and we took, if there wasn't climate data in the paper, we looked for local weather stations and we took climate data from that. And if we couldn't find approximately close weather station, then we didn't use it. And then for the UK study sites, we have a UK government organisation called the Met Office, who have a very good network of weather stations, some of which are really very, very close to our research sites. So that's the data we use rather than our own data loggers, so that's more consistent. For the summer climate data that I'm going to present and the winter climate data, those are averages of 3 months for both period. And then to measure the mycorrhiza, annually we took root samples and assess them under a light microscope. Okay, so the UK study sites, we only had 5, because the majority of our sites in the UK are Tuber borchi and Tuber estiva munchinatum. Melanosporum, traditionally we don't think it's really going to work there, but we've got some sites where we just have small numbers of trees for this very purpose for research. We have 3 in a county called Devon, which is right in the southwest tip of England, which is very mild, very, very wet. Then we have just north of there in South Wales, and then our final site is towards what we call the Midlands, which is actually very dry and quite different climatically. All the data was sampled between 2010 and 2013, and all the sites were amended for pH. So they all started off with very low pH levels, sub-6, in fact, the majority of the sites here, and they've been elevated to not optimal levels for Tuber melanosporum, but optimal levels for estiva munchinatum, so around about 7.5, 7.6. So just bear that in mind as well. And interestingly, we have a brûlée development on one of the sites. So looking at the difference in the climatic considerations between the sites in the UK, this is how much variation we get. For the summer and winter sunshine hours, so you can see that the site with the lowest sunshine hours within the study period achieved just 68% of the sunshine hours of the maximum site. And then on a per annum basis, it was 83%. So there's quite a big difference in sunshine hours. When we look at precipitation, it's even more marked. And you know, the UK is a really small country. When you compare it to Spain, we've got really big difference in rainfall levels. So here, the lowest site, this is a maximum minimum graph, was achieved 525 millimetres of rainfall in its driest year. And the wettest site had rainfall levels over 860 millimetres higher than that. So you know, it's nearly a threefold difference. So there's a big difference in rainfall. And then in terms of temperature differences, this is one of our trees actually. So the majority of the sites are Quercus ilex, and then we also have some Corylus avellana and Quercus robur as well inoculated. So for the winter temperatures, there's quite marked difference of 5.8 degrees. Summer less pronounced, just 1.7 degrees. So the biggest difference between sites is in winter temperature, not summer. And then on an annual average, it's about 5.6. So we've got big difference in sunshine hours, not much in summer temperature, and big differences in precipitation. Okay, so this graph here, the black dots are the historical data I mentioned, so the data we've amalgamated from what's published already, and the crosses are our UK sites, and this is summer rainfall versus winter rainfall. And as you can see here, there's actually there's no discernible difference really between our UK study sites and what we've gathered from the data of the kind of rainfall patterns overall. When we look at annual temperature and sunshine hours, it's a little bit different. We've got quite a distinct cluster here for the UK sites. And what this shows basically is that annual temperature, again, not much difference between the UK study sites and those naturally producing sites, but sunshine hours, significantly lower. So that's really one of the areas where we're struggling in the UK. And then when we look at winter temperature and summer temperature, again, a very nice clustering. And interestingly, not so much difference in winter temperature, but this is an average figure we're working on, the average winter temperature. The summer temperature, there's quite a difference. So the two big areas where we're struggling in the UK in terms of trying to produce melanosporum are summer temperatures and sunshine hours. Everything else, there's quite good overlaps with wild producing sites. So from this, if we're going to use mycorrhization rates as a proxy for the fruiting ability of Tuber melanosporum in the UK, we could theorise that sunshine hours and summer temperature are going to be the two constraining factors. So what we did is we ran about 9 different climatic parameters, and we correlated those with mycorrhization rates, so we could see actually how that sits and how that works in these suboptimal conditions. And what we found actually, in 8 of these we had no correlation whatsoever, but the one where we did have correlation was in winter temperature, which based on the other data doesn't make a whole lot of sense, but I'll explain this a little bit in a minute. So everything else within the suboptimal conditions, we can't really see any relationship to these climatic variables, despite the fact, for example, annual rainfall hugely differs between the sites. This is for winter temperature and mycorrhiza index. As you can see, it's quite a loose relationship, but it's statistically significant. And on our lower sites, we've got very, very, very low levels of mycorrhization. You know, it's almost going. Whereas on the higher sites, it's not too bad. It's not too bad at all. And on our sites overall, we have 5 sites, 2 of which are showing an increase in mycorrhization year on year, which is very promising. 2 of which are showing a decrease, and 1 of which is static. So we've got a completely even spread. So I've got about just 30 seconds left, so I'll sum up. That the majority of analysed climatic variables show very nice overlap with the UK and these other naturally producing sites. Sunshine hours and temperature are the two big differences. However, the only really big variable which shows a correlation in these suboptimal conditions with mycorrhization rates, and what's really driving down mycorrhization rates, is winter temperature. And although winter temperature averages are quite similar to those areas within Europe, what we have had in those specific years that we've been monitoring this are extreme cold winter events. So the year before this winter, we had -18 on several of the sites at several periods. So we had real extreme cold events, and I think it's probably that's driving down the mycorrhization rates. Also, if you remember, our summer temperature variables aren't too different. There's only about 1.7 degree difference. Whereas in the winter, it's about 5.6. So that's probably another comparison. And there's lots of other comparisons I can make, but probably not in 10 minutes. So thank you. Thank you for listening.
Thank you, Paul. With the next talk of Christine Fischer, we are turning back on tuber melanosporum soil mycelium and on its effect on soil fungal communities.
Okay. You escape this. Yeah, might as well have some. Oh, there. See. Need some water. Thank you very much. Good afternoon, everyone. It's my pleasure to present our work that focuses on the evolving soil fungi community associated with the establishment of black truffle plantations over 20 years. And these are sites that were established in areas that had intensive agricultural use over quite a long history. This is also work that was carried out by Liu Bing as part of her doctoral thesis, and I'm sure she'd love to be here today. Okay, why look at the below-ground fungal diversity in these plantations? Well, we're interested in this diversity because we would like to better understand the evolution over time of what happens below ground. We know that tuber melanosporum does not complete its life cycle in a vacuum, that it has evolved, co-evolved with its symbiotic hosts in the Mediterranean climates, but also with animals, dispersing animals, insects, and multiple microorganisms. So we want to look more at the fungal diversity over time. We had 2 basic objectives here. The first was to see if we could estimate fungal diversity in the developing burns as the plantations develop, and also to assess for changes in the fungal community as the plantation matures. This study is a small-scale exploratory study in order to take the results and design a more comprehensive and larger study in the future. The experimental design and soil sampling for this study was similar to the study that Carlos Colinas just presented. All of the soil samples were taken from the same experimental design here in plantations in Teruel. There were 6 ages, ranging from 3 to 20 years old. In this study, we had 1 repetition for each age class, and each repetition was from a different plantation following the chronosequence design. For the diversity study, we just used the soil samples at one distance, 40 centimeters from the trunk of the tree. But the sample included 3 subsamples that were pooled to do the diversity study. We began the study by extracting total DNA from the soil samples and then doing an amplification with the universal primers ITS1F and ITS4, followed by purification, TA clone technique, sequencing, and then a blast search to get our data. From the first gels that came from amplification from these universal primers, we saw a lot of mixed bands in the range between 500 and 900 base pairs, this confirming our assumption that there were many fungi in the soils at 40 centimeters from the trunk of the tree. This first lane represents a truffle sporocarp, and the other lanes represent soils from 3, 5, 7, 10, 14, and 20-year-old plantations. The sequence data was read and edited with Sequencer 4.0. All the chimeric sequences that were a result of PCR recombination were removed from the data set prior to analysis. The operational taxonomic units with 97% pairwise similarity were clustered with CAP3, and the NCBI and UNITE databases were used for blast searches. We used the Shannon index to estimate fungal diversity in our plantations. Here's a look at our overall taxonomic richness. We generated 6 clone libraries corresponding to each age group of the plantations. We had a total of 480 sequences, and after removing the short reads, the chimeras, we had a total of 228 colonies sequenced. In the 228, 62 were singlets. These were sequences that only occurred once in the database. And we had 27 contigs and a total of 89 operational taxonomic units. In our results, we had 97 sequences of tuber melanosporum in the 228 colonies. There was no detection of any other tuber species from these soils. And surprisingly, we had a very low representation of vesicular arbuscular fungi. Of the we were able to identify to the species level 68% of our sequences and 92% to genus level. There was a very significant negative relationship between diversity and age of the plantation. Here I'd like to point out again that the tree chosen to sample at 3 years old, there was no burn development at this age group. We had developed burns at 5 and 7 years, but there was no production. And at 10, 14, and 20 years, we had well-developed burns as well as good production. So with increasing age and increasing developed burns, we have decreasing diversity in our plantations. This graph shows the proportional distribution of fungal phyla that were sequenced. This is arranged, Okay, I'll try again. This is arranged according to increasing age on this side. The dark blue color represents Ascomycota. And in these truffle grounds, at 40 centimeters from the trunk, the Ascomycota clearly dominated our sites. The number of Basidiomycota sequences were low numbers, but they were stable throughout the different age classes. And the Glomeromycota showed up in 10 years, Aglomus irregulare, and again at 20 years, Scutulaspora. This graph shows the proportional distribution of sequences for the 6 most common genera. In the 3-year-old tree, the most common and dominant fungus that we found was Fusarium. There were 26 sequences from different species of Fusarium out of nearly 50 sequences from this age class. That was followed by Phoma species, and there were 4 OTUs of Tuber melanosporum in the 3-year age class. By 5 years, Tuber melanosporum was dominating the site, and that pattern increased up through 20 years. So what we saw was, as the host tree matures, we had an increasing number of Tuber melanosporum OTUs in this site, and a concurrent decrease in the saprophytic and pathogenic fungi, such that by 20 years old, or 20-year tree, Fusarium was represented by only 1 of the 31 OTUs, and Phoma was non-detected at 10 years, and Tuber melanosporum represented 22 of the 31 OTUs at 20 years. So this pattern that we observed, and even though we had only 1 repetition for this, was pretty interesting. It brought up a lot of questions for us to see a decreasing in fungi that are capable of causing plant diseases with an increasing of OTUs of Tuber melanosporum. And it brings up the question, is this pattern that we see evidence of plant protection associated with the symbiosis with Tuber melanosporum? Is this a kind of niche construction? Or is this a pattern representative of moving from abandoned agricultural lands to the development of oak woodlands with an introduction of an ectomycorrhizal fungus? So we wonder, if you took a pine seedling inoculated with Lactarius, would you see a similar kind of change in the soil fungi, the community? The other question is the diversity of fungi in the collection that we sequenced, was it distorted at all by the primers that we used? So on the studies, this study and the results that we got from this study, our next study, we would first of all increase the number of plantations that we sampled and increase the distances from the tree to see if there's an evolution not only in time but also in space with the fungal community. We'd also like to include samples from non-disturbed agricultural soils of the area to have more of a starting point at a 0 point before there was any changes in soil tilling and preparing the soil to actually establish the truffle plantations. We will apply a pyrosequencing technique so that we can process a lot more samples and with a lot more efficiency, and use the new ITS primers to reduce the biases that are associated with species with longer amplicons. Another thing we would like to examine is the Tuber melanosporum genotypes that are present in the burns. We'd like to know if there are changes in within Tuber melanosporum with the different genotypes. Are there dominances of different haplotypes over time and within different areas of the burns? Well, thank you very much.
Thank you, Christine. The next speaker is Herminia de la Varga of the University of Saragossa, speaking again on Tuber melanosporum soil mycelium and mycorrhizas in ex novo plantation.
Well, buenas tardes a todos. Good afternoon, everybody. I'm Herminia de la Varga. I'm not coming from the University of Zaragoza. I'm coming from the IRTA in Cabrils. And in the next 10 minutes, I'm going to talk to you about the persistence of tuber melanosporum mycorrhizas and extra-radical soil mycelium in a de novo plantation. As most of you may know, one of the doubts when establishing a plantation in a place where truffles have never occurred before is the doubt if the fungus will persist on the plant and if the soil mycelium will expand through the soil. Usually, to know if this happens, the methods used are to extract some plants and evaluate their mycorrhization status. We thought that not to use these destructive methods, it could be useful to detect and quantify the extra-radical soil mycelium by using molecular techniques as the real-time PCR that explained it before Xavier Parlade. So the objective of the work was to evaluate the persistence of the fungus on a new plantation. To reach these goals, a new worker was established in spring 2010. It was located in the Oriental Pyrenees in Catalonia at approximately 1,000–2,000 meters high and occupies 0.25 hectares. It was planted with 242 nursery inoculated Quercus mycorrhizal with tuber melanosporum. The initial mycorrhization percentage was around 20%, and interspersed among these plants, 14 non-inoculated plants, the red ones, were also planted. In the image, you can see how the orchard was distributed. The black ones are the plants remaining on the final plantation with a spacing of 5 per 5 meters, and the green ones represent plants to be sampled. In different colors, I don't know if it's well seen, are plants that were dead at the end of the experiment or disappeared, and the different sampling times that were done every 6 months since the establishment of the plantation. Climatic parameters available from a weather station located near the plantation were also used to establish any relation with the analyzed variables. The annual weather conditions during this period are shown in the image. No exceptional values were observed, and the orchard was established in a flat land, and the soil was characterized by a clay loam texture, moderately alkaline soil, and high content on organic matter. As I've said, the sampling was done every 6 months. In each sampling time, we sampled mycorrhizal plants, control plants, and soil from each sampled plant. In autumn 2010, after 6 months of establishing the plantation, 10 mycorrhizal and 5 control plants were sampled. Next, in spring 2011, 10 mycorrhizal plants and 6 control plants. And finally, in autumn 2011, 8 mycorrhizal plants. No controls could be available to collect because they were all dead or disappeared. To evaluate this mycorrhization, plants were examined under a dissection microscope, and some of these mycorrhizal tips were also molecularly analyzed to be sure that we were having tuber melanosporum ones. Soil was collected from each sampled plant with a soil borer by collecting 4 soil samples at 5 centimeters from the trunk that were mixed together to have a unique soil sample per plant. At lab, the soil was let to dry in order to remove any humidity excess, sieved and homogenized. After that, soil was extracted from 0.25 grams of this soil and then analyzed by quantitative Tachman real-time PCR with the specific primers recently published by Parlade et al. In mycorrhiza. To refresh you, Xavier has explained it very well how this technique works, but we measure the fluorescence emitted by the PCR when we duplicate the DNA that we want to know. So, to transform this fluorescence into milligrams of TUBER melanosporum per gram of soil, we constructed a standard curve by mixing a known amount of fresh truffle with soil. The mixture of this truffle and soil was extracted for DNA, and some tenfold dilutions were done to obtain the curve in which then we related all the analyzed samples to know the quantity that we had. So, after processing all samples, what we saw was that it was able to detect the mycelium present in soil from the first sampling, just after 6 months of the establishment of this plantation. And as you can see in the image, the quantities were increasing a long time. But unfortunately, maybe because we had a little number of samples, this wasn't statistically significant. To make you an idea, the minimum quantity detected was around 4 micrograms per soil gram in the first sampling in autumn 2010, and the maximum 14 milligrams per soil gram in autumn 2011. With mycorrhiza, we saw that the fungus persisted in all the collected plants. As you can see, the initial mycorrhization percentage was around 20%. That was maintained after 6 months, and after 1 year, we had 3 times this mycorrhization. And at the end, the mycorrhization was the double that at the beginning. When trying to establish any relation between both parameters, mycelium and mycorrhiza, we saw that in spring, after 12 months of establishing the plantation, when we found the maximum number of mycorrhiza, there was a positive correlation between the amount of soil mycelium and this number of mycorrhizas. Sorry. This correlation was not found when all samples were analyzed together, and neither when only autumn data were analyzed. After that, we tried to relate these results with climatic parameters, and we saw that there was no correlation between mycelium quantities and weather conditions, but some of the parameters were correlated with mycorrhiza. The correlated parameters were min and maximum temperature, precipitation of the previous month, radiation, and evapotranspiration. In the table, you can see the correlation coefficients. All of them are around 0.4, maybe because these parameters are all correlated within them. So, finally, with the results that we saw in this orchard, we can say that TUBER melanosporum persisted in all the radical system of the plants collected, almost after 18 months of the establishment, that soil mycelium could be quantified at 6 months after establishing the orchard, that this soil mycelium was increasing with time, as we saw in the image, and there exists a correlation between this mycelium and the mycorrhizas in spring. And finally, that mycorrhiza seems to be more affected by these climate variations almost in these first 18 months of a plantation. So we can say that Tachman real-time PCR is a good technique for the traceability of Tuber melanosporum without disturbing the host trees. So it allows us to detect the persistence of the fungus and its expansion through the soil without disturbing and without destroying any of the trees. And that's all. Thank you for your attention.
Thank you, Herminia, and sorry for my mistake.
No,
The last two talks of this session are on tuber melanosporum genetic diversity. And the first talk is of Ivan Garcia Cuncillos, of University of Saragossa now. It's correct now, I think.
Buenas tardes a todos. Mi nombre es Iván García y voy a presentar el trabajo titulado Population Genetics of the Westernmost Distribution of the Glacial Sarvival Black Truffle Tuber melanosporum, realizado junto con los doctores Sergio Sánchez, Juan José Barriuso y Ernesto Pérez Collazos. En este estudio se estudia por primera vez la genética poblacional de la amplia distribución de la especie Tuber melanosporum en la península ibérica. Mencionar brevemente los antecedentes a la temática en esta especie. Por una parte, el trabajo de Bertal y colaboradores detectaron una baja diversidad genética y prácticamente ninguna estructura genética entre las poblaciones. Por otro lado, en el trabajo de Murat y colaboradores, que utilizaron RAPID y secuencias ITS, detectaron una elevada diversidad genética, así como una estructura genética del 19,9%. Finalmente, el trabajo de Richoni y colaboradores, conducido mediante AFLP y SSR, detectaron igualmente una alta diversidad genética y una estructura del 15%. Así pues, los objetivos del presente trabajo van a ser determinar la diversidad genética, estructura genética y relaciones genéticas entre individuos de las poblaciones estudiadas, así como inferir posibles procesos evolutivos que hayan podido modelar esta diversidad y estructura encontrada. Para ello se muestraron un total de 23 poblaciones localizadas a lo largo de la distribución conocida actualmente en la península ibérica, en las regiones de, podríamos llamar, Prepirineo, Sistema Ibérico y Alto Tajo y sistemas subbéticos. La técnica molecular elegida para desarrollar estos objetivos han sido los Intersimple Sequence Repeats, unos marcadores moleculares neutros y dominantes que han demostrado ser altamente eficientes en estudios genético-poblacionales y de historia evolutiva en diferentes especies. Sí, en primer lugar, presentamos los estudios de la diversidad genética, estudiados mediante los índices de diversidad genética de Ney, Shannon y estimación bayesiana de la propia diversidad mediante el software Hickory. Como vemos, los valores totales de diversidad encontrados son valores altos para todos los parámetros. Por ejemplo, si comparamos con otros estudios realizados sobre especies del género Tuber, vemos que son superiormente más superiores. Y por otro lado, si comparamos con otros estudios sobre la misma especie, podemos ver, pues, por ejemplo, en Richoni y colaboradores, obtuvieron una diversidad genética de 0,895 utilizando el índice de Shannon, que como vemos es un valor algo superior al encontrado en el presente estudio, si bien los AFLP han demostrado ser más polimórficos que los ISSR. Por otro lado, en el trabajo de Murat y colaboradores se obtuvieron locis con heterocigosidades superiores a 0,5, que serían valores entre los que nos encontramos en el presente estudio. Un poco las diversidades por poblaciones, vemos que encontramos poblaciones altamente diversas tanto en localizaciones meridionales como en las que están marcadas en amarillo, como en localizaciones más septentrionales que están marcadas en verde. Entonces, las conclusiones de diversidad genética serían una alta diversidad genética de las poblaciones ibéricas estudiadas, lo cual es indicativo de un buen estado de conservación de la especie. Esta alta diversidad se explica parcialmente por la amplia distribución de la especie en la península ibérica, el eficiente método de dispersión de la especie, la propia historia evolutiva de las poblaciones estudiadas, y luego, por otro lado, valores obtenidos de esta diversidad genética son indicativos de una especie con un tipo de reproducción de tipo crossing over o heterotalismo, como se demostró y corroboró en los artículos anteriormente citados. A continuación, la estructura genética se estudia mediante dos métodos. El primero de ellos entendiéndola como la repartición de la diversidad genética entre las diferentes poblaciones o rangos geográficos. Como vemos en este estudio, se obtuvo una alta estructura genética del 47,89%, superior a la mayoría de las otras especies del género tuber estudiadas hasta la fecha. Igualmente, comparando estos resultados con las poblaciones, digamos, francesas e italianas, es igualmente muy significativamente superior. Entonces, dado que condiciones ecológicas, requerimientos de hábitats, etcétera, son similares en la distribución natural de la especie, hace pensar en una diferente historia evolutiva de las poblaciones ibéricas estudiadas en este trabajo. En segundo lugar, la estructura genética se realiza mediante análisis de inferencia bayesiana de tipo structure. Como vemos, el total de las 23 poblaciones se divide en dos grandes grupos. Un primer grupo sería los marcados en verde. Se corresponderían con poblaciones de localización norte, más septentrional, considerando como una barrera que separaría a estas poblaciones del resto al sistema ibérico. Entonces, dentro de los grupos del llamado grupo noreste, tendríamos por un lado las poblaciones del Prepirineo que se separarían del resto. Entonces, finalmente, este grupo noreste estaría compuesto por dos unidades genéticas diferentes entre las poblaciones del Prepirineo y, por otro lado, 4 unidades genéticas diferentes que se corresponderían con poblaciones de las provincias de Navarra, Castellón, Soria y Guadalajara. El segundo de los subgrupos, el marcado en rojo, se correspondían con poblaciones de localización más meridional en la península ibérica, teniendo como referencia el sistema ibérico como una barrera que separa ambas. En este caso se diferenciarían, por un lado, las 3 poblaciones más meridionales estudiadas, las que están señaladas en verde en el segundo gráfico, que se corresponderían con la provincia de Jaén. Por otro lado, se distinguirían otras dos unidades genéticas. Por un lado, una unidad genética correspondiente a las poblaciones de la provincia de Murcia, serían AB1 y AB2. Y por otro lado, una última unidad genética diferente estaría compuesta por las poblaciones de Cuenca, Albacete y una población de Castellón. En un principio puede llamar la atención el agrupamiento de estas 3 poblaciones en una misma unidad, pero sin embargo todas las poblaciones se encuentran separadas por menos de 100 kilómetros de distancia y no existen grandes barreras montañosas que pudieran aislar o incomunicar estas poblaciones. Así pues, concluimos que las poblaciones ibéricas tienen una característica elevada estructura genética, corroborada posteriormente con el escaso o nulo flujo génico encontrado, y por otro lado todas las unidades genéticas encontradas que acabo de mencionar. En tercer lugar, el estudio de relaciones genéticas entre individuos se realizó mediante árbol de neighbor joining. Vemos en primer lugar un gran agrupamiento de los individuos que componen cada población o unidad genética, lo cual también es indicativo de una alta estructura genética. Por otro lado, vemos que las poblaciones se vuelven a dividir en 2 grandes ramas. Una rama estaría compuesta por poblaciones del grupo noreste y otra poblaciones del grupo sureste, de la misma manera que detectaba el análisis anterior. Aparte de esta gran diferenciación de 2 grupos noreste y sureste, se detectó también que las poblaciones más meridionales estudiadas, las que hemos dicho de la provincia de Jaén, se observan altamente aisladas del resto de las poblaciones estudiadas, lo cual hace pensar también en un aislamiento de estas poblaciones que podría corresponderse con la presencia de los sistemas subbéticos entre ellas. Y por cuarto y último lugar, la historia evolutiva de la especie Tuber melanosporum ha sido descrita a partir del sufrimiento de un proceso de cuello de botella en la última glaciación. Esta teoría se basaría en encontrar poblaciones más diversas en localizaciones más meridionales y en la reducción de la masa forestal de las principales especies simbiontes en esta época. Todo ello habría relegado a la especie Tuber melanosporum a las regiones más meridionales de la cuenca del Mediterráneo, y posteriormente, con la llegada del periodo interglacial, se habría producido una rápida expansión hacia el norte. Como hemos visto, en el presente estudio se encuentran poblaciones altamente diversas tanto en localizaciones meridionales como septentrionales, lo cual hace pensar en toda una serie de poblaciones que hayan podido actuar como refugio en España, en la península ibérica, durante la última glaciación, y que posteriormente se hayan expandido no necesariamente hacia el norte, sino más bien en función de la disponibilidad de los suelos calcáreos. Se ha observado que en periodos fríos se produce un máximo de fructificación de trufas, lo cual, sumado a climas secos como sería el Mediterráneo, podría haber dado lugar a una congelación de las propias trufas de los individuos sin sufrir daños y se habrían conservado y resistido. Luego, posteriormente, con la retirada del periodo glacial, podrían haberse expandido. Esto no significa que todas las poblaciones que hemos estudiado hayan sobrevivido. Hemos visto el caso de las poblaciones de Cuenca, Castellón y Albacete, que conforman una única unidad genética con baja estructura entre ellas. Según los resultados de diversidad genética, cabe posible pensar que a partir de Cuenca se hayan podido expandir, o sea, Cuenca habría actuado como un refugio en la última glaciación y se hubieran expandido posteriormente hacia las poblaciones de Albacete y Castellón. Entonces, los resultados obtenidos concuerdan con la teoría del proceso de cuello de botella, en el que toda una serie de poblaciones podrían haber actuado como refugio en la península ibérica, no restringiéndose exclusivamente a las más meridionales. Muchas gracias.
Thank you very much. We are arrived at the last talk of today. Is Thibaut Payen of Era of Nancy.
Okay, so first of all, I want to thank the organizer of this conference to allow me to present my work. So I studied for my thesis the polymorphism in tuber melanosporum by genome resequencing. The introduction will be quite redundant with the precedent talk, but my study is quite complementary. So, as most of you know, the Perigord black truffle production have decreased a lot during the last century. A lot of reasons for that, like the 2 world wars and rural desertification, human factors, and more climatic factors like landscape changes or climatic changes. So, a lot of studies have been done about the impact of the climate change on tuber melanosporum. One paper, quite recent, in last December, have linked the decline of the production with the dwarf, and in 2008 there was already some conference about the impact of climatic change. So, what can a species do to adapt to such an important thing as climate change? So, it can migrate. So, for example, for tuber melanosporum, it can adapt. It can go to into the north with more climatic suitability for the species. It can adapt, and this depends on the genomic diversity, the way it reproduces, and in general the plasticity of the genome, or it can fail to adapt and disappear. We don't want that, but that can happen. So, like presented in the precedent talk, the genetic diversity in tuber melanosporum have been debated since 15 years. I will not say the content of this diapositive because you know that have already been presented. But so my study is very complementary with the precedent one, which was studying a very small part of the genomes, but on a lot of species. On my side, I have few strains. I have only 7 strains, but I study the whole genome. So I want to know if the things found in genetic studies are representative of the whole genome. For that, I used the reference genome, which have been sequenced in 2007 and published in 2010. We know that tuber melanosporum is a very big genome, but which have a very small set of genes and a huge quantity of repetitive sequences. So, the methodology of my study: first, we have sampled some strains. We have extracted the DNA and sequenced it, so the whole genomes, and assembled it to have the new genomes and study the polymorphism of the complete genome. So, on the sample part, we have selected strains to be the more different possible, with strains from the south of the natural area in Spain and from the north of the natural area in Alsace in France. From this sample, we have obtained quite a good coverage of the genome. More than 97% of the reference genome have been covered, and you can see, 120 millions of the 125 millions of the reference genomes have been found in the 7 strains. We consider that as the common genome of the species, and on the 496 genes, only 15 were missing in some genomes. So after that, we have studied the polymorphism. We have focused on SNPs, single nucleotide polymorphism, which is the transformation of one nucleotide in one other. And we have found quite a different number of polymorphism in different strains. And the total non-redundant was more than 500,000 SNPs, which is one every 240 base pair. So, that have been presented in the previous presentation, there are polymorphism in microsatellites, but the question here was where are the polymorphism in the genome in general. So we can see most of them are like expected in a transposable element and other genomic regions, but we can find some polymorphism even in genes. So the three first part here, even more, in exons which are coding for the proteins. They have a very low level of polymorphism in comparison to other parts, but they still have. So we have done a small tree, very less impressive than the precedent one. But still we can find that we have a geographic structuration of the population with the two of Spain which are together, the two of the north of France together, and the three of the southeast of the area together. One other thing, we wanted to confirm or infirm the results found in genetic studies. So we have compared the number of SNPs in Tuberculosporum on the other species. Where we don't have a lot of species to compare to, because that's quite a new field. But we know that Tuberculosporum is more polymorphic than the two other filamentous fungi which have studied of this kind, and less than some other fungi and or plants. But yes, still the study from 15 years ago is quite wrong. So to conclude, we have resequenced 6 new Tubermelanosporum genomes. That's the first project of this kind in ectomycorrhizal species. We have found what we consider as a pangenome, what we consider as a core gene, and yes, we have confirmed mostly that Tubermelanosporum is a species with a high level of genetic diversity. What I haven't presented there because of the only 10 minutes is that once a little less than 1/3 of the genes have SNPs in their coding regions, so there are differences in the protein in Tubermelanosporum that have to be studied more carefully. And we have started, but that's not totally finalized. We can search for adaptation signature that will be presented a little tomorrow by my supervisor, Claude. So I want to thank the people which have gave the sample and my colleague and supervisor for this work, and thank everyone for your attention.
Thank you very much, Thibault. And I would like to thank all the speakers of this session for the high quality of their presentations. Now we have time for some questions, so please, your turn. One question?
Hello, my name's Stephen Langrell. I have a question for last speaker. I very much appreciated your presentation, and I just wanted to ask about these transposable elements that you were mentioning, and I think you were giving us a little bit of a taste of what might be coming tomorrow. But looking at the sequences, the whole genome sequences that you've got so far, and I appreciate you might not have had the time or opportunity to perform any other type of experiments. But looking at your data, are any of those transposable elements that you see, that you can read and analyze on the screen, do any of them look active on our drivers for the diversity or some of the diversity that's been discussed this afternoon?
Yes, we have started to study the different families of transposable elements. Some have very high diversity between elements of the same family, which is a signature of a very old family, but some are closer, so yeah, more recent family. Active ones, that's kind of difficult with this kind of study to be sure of it, but there's recent ones. Active one, not sure.
Yes, on these questions, so in the genome of Tubermelanosporum, we identified an old colonization by retrotransposons, and we identified some elements that are expressed during the fructifications. So we have not yet identified active elements, but we know that some of elements have their protein, like transposase or reverse transcriptase expressed. But we have no evidence that some of them are active and can jump and modify the genome.
Sorry, thank you, Chairman. I have another question, if I may. This one's, well, it's more of a confirmation. If I understood correctly from Christine Fisher, she's over there, I think. I really appreciated your presentation as well. I found it amazing. I'm quite interested in the variation in the amplicons you get from the first round amplification with your universal primers. So have I understood correctly that what you did then was you cloned a subsample of your amplicons, and then have I understood correctly as well that basically you just made a subset or a subsample of clones for sequencing? What I'm really getting at here is, have we really evaluated the true richness of the diversity that you were presenting, or are we looking at an underrepresentation, or is the richness much greater based on like a bottleneck in the approach? Maybe I misunderstood something. Could you possibly clarify?
Let me see if I get your question. Are you asking about really the diversity? What kind of diversity did we find?
Yeah, basically, if you use your universal primers, you get a whole, a wide range of different.
You get a lot of fungi.
And then you clone.
Right, but whatever you get you can't, from your cloning process, there's always some error that you introduce. There are some colonies that you are more successfully cloned than others. So you have some error at that level when you're looking at total diversity of all the fungi that are extracted from the original soil sample. And then, depending on the primers that you use, you have some preference for the sequences that have longer amplicons. So those are some changes that are coming up with recent developments in looking at what the universal primer used for amplifying fungi. But we, nonetheless, in the 3-year-old plantations, the total number of sequences or operational taxonomic units that came out was much higher than what we found at 20 years. There was a significant decrease in the number of actual taxa that we encountered. There were almost 20 different taxa in the 3-year-old, and there were closer to 12 or 16 by 20 years old. So there was definitely a decrease. But what I was surprised by was that there was not a lot of diversity in these soils. We didn't see a lot of Cenococcum, we didn't see AD type, we didn't see a lot of the ectomycorrhizae that we normally find in truffle beds. Of the Basidium mycota, we found Hysterangium, we found Cryptococcus, but we didn't see a lot of the other fungi that we typically would find. And we think it's due to the tillage practices and the history of these areas that have been agricultural lands, abandoned lands. I don't think there was a lot of diversity in these soils to begin with, which is one reason why we'd like to increase the study, use a lot more sites. But what really was interesting was how quickly we had a dominance of Tuber melanosporum, and that maintained over 20 years. So we asked the question, how much diversity then is there within the tuber population itself, and does that shift? So.
Other questions? No? No questions? Oh, one question.
Buenas tardes, yo soy Joaquín Doñate, soy truficultor y quería hacer una pregunta casi para todos los científicos. Yo tengo un tema que nos inquieta bastante a todos y es saber diferenciar ciertas variedades de trufas. Por ejemplo, cómo diferenciar bien una indicum de una melanosporum. Me gustaría saber si el campo científico está investigando algún tipo de reactivo que sea fácil y rápido, que no tengamos que tener un sistema molecular para poder saber qué trufa es de una especie o qué trufa es de otra, y si se va a poder hacer algo al respecto. Gracias.
Voy a responder yo mismo. Es una buena pregunta realmente, me parece que sería interesantísimo el que tuviésemos un reactivo de testaje inmediato. Que yo sepa, en donde lo que trabajamos nosotros solamente en molecular estamos intentando solucionar este tema. No creo que sea tan fácil, so pena que el equipo de la Universidad de Zaragoza, lo que a mí corresponde que conozco, conociendo los diferentes elementos volátiles, sea capaz de diferenciar unos de otros. No, en nuestro caso sería una cosa interesante y es una línea de investigación muy curiosa, muy buena, pero un poco verde todavía.
Okay. If you haven't thought, you have 5 minutes again? Okay.
I'm not sure if I understood your question. So you want to know where are you speaking? Who is that? You want to know if there are methods to distinguish tuber melanosporum from tuber indicum. Is it true? Did I understand the question? Yes, there are specific primers. You can use them, and it is possible to distinguish them. Not from the morphological point of view, but we have done many publications about this. One is from Murat here, and there are many, so it is possible. Without molecular, you say, from the mycorrhizal point of view, I'm afraid no. But in the fruiting bodies, I think if you are an expert, you can. They are specialized people. No?
If you don't have other question, another question here, Claude. Question or answer?
Yes, I have a question for Spanish groups. Do you have investigated the introduction of tuber indicum in Spain? Someone already screened some truffle ground?
Well, it's not that we're really investigating it. You know, if we have a chance, we look for it, and so far, no hits.
In French too, for the moment we never found.
Okay, in CTFC, when we evaluate seedlings for plantation, routinely we check for indicum.